short answer format for the rest of it. Okay, and so the plan is to make it a mixture of things. It's not going to be so mathematical as it is going to be really testing your understanding of concepts and how to apply them, right? So the questions that I ask in lecture are often of that nature, right? Where I sort of ask you to think about the concepts and really think about like the applications of the concepts and what that means for the biology. So that's the kind of question that you should think about having on the exam. Dr. Camera actually had some of those types of questions. She and I went over a list of questions that she had generated and that she went over in recitation sections. So those kinds of questions where you're really thinking, what does it mean that you have this type of interaction? Like, what does that do to the structure of DNA? What does that do to the structure of a protein? You know, what are these forces doing? You know, what does it mean that your pKa is this versus that, right? I'm less interested in exact numbers on buffers, right, than I am in really having you understand what the fundamental concepts are. Which doesn't mean that there will be no numbers, but there's not likely to be really complicated mathematical questions as much.
The weekly quizzes, is that kind of on par with the exam
Well, the multiple choice questions, yes. With the short answers, obviously not, because you can't really do a short answer. But yeah, I mean, the kinds of, the multiple choice questions are going to be like the quiz questions. I mean, I make the multiple choice questions on the weekly quizzes, and I'm making the exam. So it's a fair bet that they will be in a similar vein. As I said, the short answer questions will be kind of applied knowledge questions, right? So think about this, apply it to that. Explain to me the difference between this and that kind of thing, okay? I'm not going to promise to give you guys a practice anything, but I will see what I can do to sort of dredge up something that at least maybe cobbles together some problems for you. So, and then I'll just sort of write down the pencil time. Okay, so the next thing I want to talk about just really briefly is a summary of what we went over before, and there are a couple of points that I want to make, and then I'm going to go over a misconception on a slide that someone caught that I think is worth sort of talking about. Although let me say the details are not necessarily so important, but I still think it's important to sort of have a basic understanding. So the previous lecture, we were really talking about nucleic acid structure, right? So that was really the structure lecture. And you just have to remember that these are always written from 5' to 3', which is really critical because it means that when you write it this way versus that way, these are not the same thing, right? This is not the reverse complement of that. Definitely not the reverse complement. This would start with an A, right, if it was. Okay, so another point, Chargaff's rule states that in DNA, the number of purines is equal to the number of pyrimidines, i.e. A equals T and G equals C. Actually, if you think about the purines and pyrimidines, it really just states that A plus G is equal to C plus T, right? So that's the actual rule. But it's sort of widely accepted to mean A equals T and G equals C as well. Okay, so you should know the key features of Watson-Crick base pairing, and you should know what Watson-Crick base pairs look like. You should know the Watson-Crick model for DNA, which is the bDNA form. You should know, you should be able to draw paired bases. So I may indeed draw this base, right? Or draw the Watson-Crick base pair 4, you know. You should know that in nucleic acids, that DNA and RNA structures are held together by hydrogen bonding, which gives specificity, and base stacking, which gives the main thermodynamic driving force. So base stacking is really critical driver of folding, right? And actually one of the main thermodynamic sinks for folding. But the hydrogen bonding is a really critical aspect of specificity. It's not the only thing involved in specificity. A certain amount of the base pairing, a certain amount of the stacking energy is also sort of aiding specificity, but it definitely is really critical for specificity. In DNA and RNA, the hydrogen bonds are cooperative. Whoops, I skipped over a step. In nucleic acids, the structures are held together. We went over that. Coordination of magnesium with the phosphate backbone helps shield the charge repulsions. So if you didn't have magnesium, you would have a much less stable DNA because you would have all this anionic charge held really close, right? And those charges do not like one another. They are repulsive to one another, right? So having magnesium, which is a positively charged cation, right? So sitting between those negative charges helps to balance that out and make it so that they are not repulsive to one another. Okay, so you get coordination of magnesium to the backbone. Again, these are cooperative for hydrogen bond formation. You should know the structures of B form, A form, and Z form DNA and the differences between them. Okay? And you should know hexene base pairing. I'm not so concerned with you necessarily being able to draw them, but you should understand what it is and where it is, right? And the idea that it's an addition to Watson-Crick. You should understand hypochromaticity in DNA. You should understand the melting temperature, what the melting temperature is telling you about the stability of DNA, and how it's calculated. And you should understand the impact of different bases and sequences on the melting temperature. And before we get into sort of the next bit of that, I'm going to go over something that actually was from the previous, a couple of lectures ago, that was a minor point that was confusing to some people. So first let me say, you do not need to memorize these numbers. Okay? So I don't care about the memorization of these numbers. My point in having shown the slide actually has nothing to do with memorization of the numbers. And the only thing I wanted you to know from this was that as you go up in temperature, the pH of water actually goes down. That was it. Okay, that was the main lesson of this slide. So all of the details are unimportant. However, someone from the class was confused about how this could be the pKW when it doesn't include the concentration of water. And so I thought I would just take a minute to say that the Kw here is indeed the equilibrium constant for water. And it is so if you remember that equilibrium constants are unitless, right? They have no units. And we never really talk about why that is in class. But the reason that is because the equilibrium constant is actually the concentration of something at the equilibrium over the concentration at the steady state, right? Or delta G is 0. And at the steady state conditions, everything is assumed, most things are 1 molar. And so therefore, it always cancels out because it's 1, right? Now, water in this case is actually 55 molar at steady state. But the water concentration isn't changing. This equilibrium is so, it's basically not changing anything about the concentration of water. Okay, so it's 55 over 55. It drops out of the equation as 1. And that's why this is the actual equilibrium constant. You could put it over the concentration of water, at steady state, but it would equal 1, and so it simplifies to this. You don't need to know all of this, but it was a little bit confusing, and sometimes when people look at something that looks really different than what they're used to, they get confused by it. So I just wanted to point out that it's because of the fact that it's not significantly different from the concentration of water at steady state that the concentration of water drops out of the equation. Okay, and the reason that other things don't is because the steady state concentration is usually 1. Okay, so anything over 1 is that number, right? So that's why. Okay, again. Okay, so we had briefly gone over this. We'd gone over the concepts for this when we talked about base stacking, and I'd shown the slide and we'd briefly gone over this, but I wanted to actually remind people of this because this is actually one of the things on the quiz that was most often incorrect. About 50% of the class did not get this right. I gave 2 different coding strands, and I said, okay, so and they had the same GC content, but they were different sequences. And I said, true or false, these have the same melting temperature. And about half the class thought it was false, and about half the class thought it was true. And the answer is it's false. And the reason is because it has to do with what I had pointed out earlier about the fact that when you have different sequences, you have different bases stacking upon one another, right? And although some of the energy in the GC base pairs and in the AT comes from the hydrogen bonding, and the networks of hydrogen bonds aren't changing, the order in which the bases are stacked is, right? So there is indeed a melting temperature difference. And proof of principle, or proof of this comes from the melting temperature of these 2 pieces of DNA. So this is a piece of DNA that's AT, right, with its complement. And this one is all A's and all T's, right? So they have exactly the same concentration of AT base pairs. But the melting temperature of the one that's alternating A and T is 48.5, and the melting temperature of the one where all the adenines are stacked one on top of the other and all the thymines are stacked one on top of the other is actually 56. Okay? And so that's a really good example of the fact that sequence matters in melting temperature. Okay, it's not just the percentage of GC. Now, if I give you things where all of them have different percentages of GC and I ask you which one's more stable, yes, go by GC content, right? But if I'm asking you if these 2 are the same and they have 2 different sequences, then the answer is no. And it's because the stacking interactions are different. Okay? So I thought it was worth going back over that, especially since people had trouble with that on the quiz. So I'm going to stop here and just ask really quickly, are there any questions from the previous lecture that people want addressed before I move on? Okay. So let's talk a little bit about how DNA is actually studied. So what are some of the things that we do to look at DNA? In particular, let's talk about gel electrophoresis. So we'll talk about other methods if we manage to get to genetics way down the line, but for now, let's just talk about some simple things. So gel electrophoresis. So in gel electrophoresis in general really takes advantage of the fact that when an electric field is applied to a solution, molecules with a net negative charge move towards the positive end, right? So they go from the negative charge to the positive charge. And so if you put DNA, which is a negative molecule, right, because it's a polyanion, into this field, it's going to migrate through the gel towards the positive end, okay? So DNA will migrate. And so the typical gel that one uses in DNA electrophoresis is agarose gel. It's literally a polymer of a carbohydrate. And so it's the media through which you migrate, and the migration is a combination of charge and things like hydrodynamic volume. So this is what a gel actually looks like when you stain it, okay? And this is actually looking at it using UV light, and it's using aethenium bromide as the fluorescent stain, okay? So again, it's typically run on agarose. The electrophoretic mobility is determined by size, but also shape. So I want to point out that what you're doing is you're migrating through a polymer matrix, right, of agarose. And so as you can imagine, like these are uneven pores all over the place. It's not just a matter of how charged you are, right, but also what shape you are. If you're a compact shape, you're going to potentially migrate faster than something that has a different shape, right? So depending on your shape, you actually get different migration through the gel. So compact shapes will move faster than things that are like loose and floppy. Okay. And so that's called the hydrodynamic volume, right? So it's not just a matter of charge, it's also a matter of shape. And you should just know that. And that's also true when we get to proteins. If you talk about protein electrophoresis, SDS page shells, it's the exact same thing. It's a combination of charge and shape. Okay? Oftentimes people think it's just the charge, but that's not actually true, right? Because you're migrating through this gel and the pores are different, right? So, okay. So typically though, you can get a pretty good sense. In general, it tends to go with the size of the DNA, especially for smaller linear fragments. And so you usually can use molecular weight bands, so molecular weight standards, where you can then calibrate DNA by size using gel electrophoresis. And this is the absolute common way to test whether or not your amplification of a gene has worked in a laboratory. So if you go and you do a laboratory on DNA, oftentimes people will have you run a DNA gel, okay, to be able to test the molecular weight and see whether you have the right size band, okay. In fact, this is done routinely in my lab. Okay, so you stain the gels with a molecule called aethenium bromide, and this is what aethenium bromide looks like right here. And aethenium bromide is a UV-active compound, and so I have a question for you. So how do you think that aethenium bromide might interact with duplex DNA? So thinking about what we know about DNA structure, how do you think it could act?
Doesn't it go in between the nucleotide base pairs?
Yeah, it's exactly right. So this actually interacts, it does what's called intercalation, okay? And that actually makes sense just looking at the physical properties of the molecule, right? This is an aromatic ring which has polarity in it, right? Polar aromatic rings, that sounds exactly like something that could have pi stacking interactions with bases, right? And in fact, that's exactly what one sees. So if you look at how aethenium bromide what aethenium bromide does, it actually swaps in between bases in the DNA and it kind of warps the backbone. It sort of unwinds the helix a little bit when it does this. It warps the helix, okay?
So is that carcinogenic?
Yes, actually that is why it's carcinogenic. It's because of this insertion property, because it's an intercalator, that is why it's carcinogenic. That's a good point. So be careful if you're using aethenium bromide because it is indeed a carcinogen. Okay, and it is something to be careful of. It's especially, even more of a teratogen, which means that it's a problem if you plan to have kids because it can cause mutations in your genomic DNA. Okay, and so that's one of the things that people just have to be careful about. It's not like at very low concentrations, but still, if you're in a lab and you're using ethidium bromide, you are careful with it. Okay, it is a dangerous chemical. So, and it's because it does exactly this, because it intercalates between the bases of your DNA. In fact, this idea of intercalation, which you see here, is something that many things do. So cisplatin actually does this as well. So there are multiple different drugs even that intercalate in DNA. Okay, and lots of DNA damaging agents are intercalators. Okay, so which cisplatin is arguably one of. So these are sort of examples of thinking about the structures of molecules and what we know about the very simple properties, right, and the things that actually control DNA structure actually tell you a little bit about some of the chemistry and biology of these molecules.
Does it somehow alter the ability of DNA to travel across the gel?
No, it doesn't alter the gels. I mean, I think it does have small structural effects, so it does affect the structure of the gel, but not to the extent where it's going to alter the migration significantly. And also, actually, I will take that back. If you were to intercalate before you ran the gel, probably, but the way that people run DNA gels is you usually add the ethidium to the agarose, so the dye is actually dyeing it as it migrates. And so usually it doesn't have much of an effect. People also will soak the gel after, although usually it's a little safer just to add it to the gel itself, which is the typical way of staining. So it might, but not enough for it to make any significant difference in the way people use it. Okay. So I also wanted to just take a minute to sort of talk about DNA and some of the things that we have found about DNA by thinking about unnaturals. So, I teach chemical biology at the graduate level, and, one of the things that we talk about is sort of how chemists are really, altering basic biomolecules to try to find new things about nature. And so it turns out that you can actually incorporate unnatural base pairs into natural DNA, and the polymerases will still accept them. And so all of these base pairs that I've shown you actually have been accepted by polymerases. And this base pair right here has actually been replicated within a living cell, okay? Using some tricks, having to put in new enzymes and other things so that the cell will accept it. But still, the fact that you can get a replicable, unnatural base pair in a genetic code is pretty incredible. And it's told us a lot about how these work. And let me just also point out, the work by Benner, these are actually now used in some of the tests that people do for viral DNA, because they actually allow, this unnatural allows for really good amplification of various other types of DNA using some tricks with the polymerases. And so it allows for really, really low copy number detection of viruses. And so I think the current hepatitis C test actually, the test for it actually uses these unnatural base pairs in it, in the ends, okay, in the polymerase reaction. So, but one of the things I want to point out about this, so this is the work of Floyd Rumsberg, who is a colleague and friend of mine, and Floyd's work is really interesting because take a look at these bases, right? What isn't in this bases? So what doesn't this unnatural base pair have? No, because it's connected through the phosphate groups, right? But look at, this is the bases. What isn't? Hydrogen bonds. There are no hydrogen bonds, right? So it used to be thought that it was required that you had to have hydrogen bonds for specificity. But it's not completely true. This actually is a shape-shape interaction. It's a hydrophobic interaction. And in fact, if you look at how, so this is actually the DGGC, and you can see the hydrogen bonding matrix. This is what Floyd, I don't know why he gives them these names, because really these are not at all easy to say names. DNAM D5SICS6. I just think, but if you look at this, it turns out that they don't have hydrogen bonding, and if you look at them in duplex DNA, they actually are intercalated with one another, and they're actually a little bit more stable because they're pi stacking. It's a surface-surface interaction. What's interesting is you would think that if they were pi stacking like this, that this would be a problem because the polymerase couldn't recognize it. But it's kind of cool because what happens is when the polymerase that actually replicates DNA, right, recognizes it, what it does is it causes an induced fit. It's a double induced fit. So the polymerase changes because of recognition of the base, and the bases change because of recognition by the polymerase. And so you actually, in the polymerase, that's what the structure looks like. It looks kind of like the Watson-Crick flat structure. So when it's not in the polymerase, it's like this. It's basically aromatic-aromatic interaction. And when it's in the polymerase, it goes edge to edge, which I think is a pretty cool trick. And this has actually allowed him to get this base pair into unnatural organisms to the point where he's now not only been able to replicate it, but also to encode unnatural amino acids using it. And that work was actually just published last October. So it's a kind of cool extension. And so just to think about, why might you want to make an organism that uses other base pairs? You know, so this is the, again, you don't have to answer this, you don't have to write anything down, but I just wanted you to sort of think about, like, why is it that we're interested in doing this, right? The thought experiment. Okay, so that's the end of the DNA section. I thought I would end with something kind of cool and really, really current about what people are doing in the DNA research field, right? So we're about to start on the 4 levels of proteins. So before we go on, are there any questions about DNA, RNA, the material? Okay. Okay, so in talking about proteins, so now we're going from DNA, and the next macromolecular class we're going to talk about are proteins and peptides, right? So you know, as we know from our if you think about the hierarchy of life, people say the genetic information flows from DNA to RNA to proteins. We kind of covered DNA and RNA in one fell swoop, and so now we're on to proteins, right? So when we think about proteins, there are different levels to think about. There's what we call the primary structure, which we think about as the amino acid code. There is the secondary structure, which are things like alpha helices and beta sheets. There is the tertiary structure, which is how these alpha helices and beta sheets come together to form the localized structure. And then, if this can be a subunit of a protein that might have multiply encoded subunits that are separate strands of protein, right? And they can come together in what we call a quaternary structure, right? So these are sort of the 4 levels of architecture. And we're going to start out in this lecture, really, we probably won't even get to the primary structure because we're really going to be talking about amino acids. So we're going to go all the way back to the very, very simple part of thinking about this macromolecular architecture. So just to point out, this is an amino acid. We're going to talk about them a lot. This entire lecture is all about amino acids. An amino acid, when they are, so they have an amine group and they have a carboxyl group, right? So this looks a little bit familiar. And they have an R group, and we're going to talk about all of this in excruciating detail. And when you put them together in amide bonds, right, and this is a good time to, for those of you who don't remember, this is a great time to review the chemistry of amide bonds from organic chemistry, because some of that actually is going to come into play and matter, right? So thinking about, you know, whether or not, you know, is this a freely rotatable bond? The answer to that's no. If you don't believe me, do the resonance structure, right? That actually has a huge impact on protein folding, as we'll get to. But so these come together in amide bonds, and this is what we think of as a peptide. This is the peptide chain. You write it N to C, we'll go over that, and here are your side chains. When you put a lot of these together, this is our primary structure. You can see right now this looks like a whole bunch of letters. You are expected to know what all those letters mean, obviously not at the beginning of this lecture because we haven't gone over them. But yes, you do need to memorize all your amino acids. They're 3-letter and 1-letter codes. Question? Yeah, we'll get through all of them, and then secondary structure, and then tertiary structure, right? So, okay. So let's talk about the basic structure of an amino acid. As I said, we're going all the way down to the very basics. So the amino acid again has an amine group, an alpha carbon. So this is called the alpha carbon right here, and the carboxyl group, right? This is the general structure of all amino acids is exactly the same. If you draw them out the way that an organic chemist would typically draw it, right? This R group, if you're going N to C, which is how you should draw it, and you draw this going up, this is coming out of the board at you, okay? If it's going down, it's going back, but that's because you're actually drawing. If you flip this, if you think about it, if you flip this over, right? If it's like this, and you flipped it over, now it's in the back, right? If this time's the R group. So just remember that. So it's gonna shift. But when you draw the amino acid like this, which is the typical way for individual amino acids we draw, this R group is coming out of the board. That's because this is the L isomer, and we'll go over that in a minute. So the general structure of all the amino acids, again, is absolutely the same. They all have this general structure where it's this R group that changes, okay? So the R groups that we're going to talk about, there are actually more than 20 amino acids that have been identified in nature. However, there are pretty much 20 amino acids that are commonly found. There are others that people have found in bacteria and in specific instances. We're not going to cover those, okay? We're going to talk about the 20 canonical amino acids, which are what we think about when we think about the genetic code. So there are 20 different kinds of R groups. We're going to go through all of them in great detail. And so again, we'll go through what all these structures are, but you're going to need to know the structures, you're going to need to know the single letters, you're going to need to know the three-letter abbreviations. This is the place where flashcards are good. This in your bases, okay? So get used to them. Okay. This is an overview of them. Again, I'm not going to really spend time on this because we're going to go over them in a lot of detail. The one thing I want to point out is, and I don't even particularly like this representation of them, but the one thing I want to point out is there are all sorts of different groups of amino acids. Some of them are aromatic, some of them are basic, some of them have sulfur or hydroxyl, they're cyclic, and we're going to go over it in detail, these groups and what they do within, so sort of what are some of the structural consequences of having these different R groups, in the context of a protein or peptide. Is there a question? Okay. Okay, so before we go into all of the different R groups, let's talk a little bit about chirality. Okay, so if you look right here, you notice that the alpha carbon, if R is something other than a hydrogen, right, the alpha carbon is a chiral center. Okay, has 4 different groups on the carbon. That makes it a chiral center. All common amino acids, so all of the common amino acids are the L isomer. So when we talk about the 20 amino acids, they are all L. Okay, you do in nature have the D isomer amino acids in some rare instances. It turns out that there are naturally occurring D amino acids. They're used very specifically for nature because they are different. And one of the places that they're now finding them are in neuropeptides. We don't understand all the implications of this. This is actually a very, very new area of research, right? Because canonically, all of the amino acids are the L amino acids. There are some D, but again, they're rare, and in the cases where we found them, they seem to have a very specific reason having to do with the stability of neuropeptides or proteins. Okay? So again, they have been detected, they do exist, but we don't see them very often, and only some of them exist. Not all of them are found in the D form. This is, again, the L isomer. If you look at L and D, you'll notice there's a mirror plane here. That's because these are enantiomers, and this is the one that we are going to concern ourselves with. All the amino acids except glycine, which has R equals H, right? So it's the one case that I said where it's not chiral, have a chiral center and are chiral. Some of them have more than one, okay? But all the amino acids except glycine have a chiral center, and glycine is the only one that is achiral. So this is another representation of an amino acid. This is alanine, where this is R is a methyl group, right? You can see if this is the L isomer. It's coming out of the board. It's coming out of the board, and this is how you draw it in a Fisher diagram. Okay. So again, they're not, just as a reminder for those of you who may not remember, right? Enantiomers. They are not superimposable on their mirror images, right? So this is Samuel L. Jackson and this is Samuel D. Jackson. Okay? And so the L isomer and the D isomer, and the L isomer is what we're going to have in amino acids. Okay? Okay, and this is, again, I don't want you to memorize this. This is not about details. I just want you to know where the L and D comes from. So this is just referencing where L and D come from. They come from the Fisher diagrams. Okay? So in the Fisher diagrams for sugars, this is D-glyceraldehyde, that's L-glyceraldehyde. The hydroxyl group in L is on the left-hand side. This is what determines the L or D position. In L-alanine, if you draw it with the carboxyl up, the amine is on the left-hand side. And that's why this is L. Okay. Again, this is just a detail. I just thought it might be nice for you to know a little bit about where the L and D comes from. Okay, so it comes from the Fisher nomenclature. Okay, and I also wanted to point out that it has consequence. So chirality has consequence in many different ways, but the fact that we have chiral amino acids actually does have consequences. So this is L-asparylserine, this is D-asparylserine. You notice that these are enantiomers of one another. They are not exactly the same. Steve Kemp's group, quite some time ago now, has actually made HIV protease, has actually synthesized the HIV protease using all L amino acids, which are the natural, and he synthesized the mirror image using all D amino acids, right? So he actually synthesized this protease in both forms. And one of the things that they found is that, so this was prepared, this is actually the paper, it was in Science in 1992, so it was quite some time ago, but the enzyme enantiomer cut only the corresponding substrate enantiomer. The chirality matters, right? You had to have the L protease, which is the natural HIV protease, only cut the peptide for the L peptide, for the peptide that was made with L amino acids. It wouldn't cut the D. And in fact, let me say that this is one of the reasons why people are interested in this sporadic use of D amino acids by nature, because it looks like sometimes nature purposefully isomerizes between the D and the L at positions where it wants to prevent proteolytic cleavage. And as I said, they've seen this in some neuropeptides. And the idea is that this may be a mechanism for your body to prevent certain neuropeptides from being cleaved and therefore destroyed, right? So I think that that's very interesting because it's actually taking advantage of the stereochemistry to alter the ability of enzymes to catalyze the cleavage of specific things. Okay, and that's what proteases do. They take a protein and they clip it up. And really, they catalyze the reaction of the hydrolysis. Okay? Okay. So that's a little bit about the stereochemistry and the fact that the stereochemistry has consequences. What about the pKa? So since we just had a talk about pH and pI and pKa, I thought we'd take a minute to talk about the pKa of the amino acids. So amino acids are typically not written like this. Okay, sometimes we write it this way if we're trying to write the neutral form, but realistically in aqueous solution, they're the zwitterion. So a zwitterion is anything that's both positively and negatively charged. So the reason that we draw them like this is because the pKa of the amine is typically around 10 in a free amino acid. This is not what the pKa is in a protein or peptide. Okay, it's the pKa of the free amino acid is around 10. Okay, and the pKa of the carboxylate in a free amino acid is around 2. Okay, so this is fairly acidic, this is fairly basic, this ends up being the zwitterionic form. Okay, and so at physiological pH, at pH 7, both of these groups are going to be ionized. Okay, so this is going to have a net neutral charge, but it's going to have a positive and negative end. Okay? Okay, and just to sort of remind you, so if we look at a peptide, so this is an example of a peptide, in proteins or peptides, only the N's and the R groups are titrated. So all of these amino acids in the middle are now in amides. These are no longer going to have the same acid-base properties, right? Because an amide, protonated amides are incredibly acidic. Or we'd be talking about, something where you would have to put another hydrogen on there and make this positively charged. And the pKa of that's going to be very, very, very low, and you're not going to see it protonated. So amides at neutral pH are neutral, right? They're not charged. But the amino terminus, which in a peptide is between 6.8 and 8, right? So notice that it's not as basic as it is in the free amino acid, right? So the amino terminus is going to be positively charged, and the carboxylate is going to be negatively charged. These charges cancel out, and really it's the side chains that are going to determine the pI of a protein, okay? So if we think about those side chains, and we're going to go over these in more detail side chain by side chain, but I wanted to give you some idea about the pKa's. Again, these numbers vary depending on the context. So this is kind of an average number that people give for pKa. It will vary depending on the context. If I ask you to calculate something, I'll give you the pKa's. But you should know the relative pKa's, meaning you should have a sense of indexing, right? Like, is something more or less acidic or basic than something else, right? And where does that sit in regards to neutral? Okay, you should definitely know that. The absolute number, I'm not so worried. Like, if it's 6.8 versus 6.5 versus 6.6, I'm not so worried, right? But you should know that, for example, that histidine is about it's around 6.5, 6.6, which makes it very close to the pKa. It makes it close to the pH of water, right? If you think about pKa and pH, that means that this is going to be about 50-50 of the positively charged and the neutral, right, in water, approximately. Aspartate and glutamate side chains, again, these are carboxylates. They have a pK of around 4, right? So that means that in water, or in aqueous solution, I should say in buffer at pH 7, these are going to be charged, right? So approximately 1,000 to 1 of the charged versus the uncharged, right? Just doing the Henderson-Hasselbalch equation, 10 to the 3. Cysteine has a pKa of about 8.7 for the protonated. So this is going to be mostly at about, almost 100 to 1 in the protonated form. But still, 1 out of every 100 or so molecules is going to be deprotonated. That's not a small amount, right, in the context of something. Tyrosine has a pK of around 10, kind of like phenol. Again, most of this is going to be in the protonated form. And now if you think about it, it's about 1,000 to 1 protonated, right?
On the quiz, there was a question that said that it was kind of like the scenario.
I know, and in that case, the trick for that was that, so it was false, but the reason was because I said each individual molecule.
No, it was a multiple choice one.
Which one?
It was like in water, pH 7, you have this amount of phenol with a pKa of 10.
Yeah.
And the
pKa was 10, and I said, what is going to be so is it all going to be dissociated? Is it all going to be associated?
No, it was 1 out of 1,000 molecules,
yes.
But you just said it there, 1 out of 1,000.
Well, that's because I'm estimating, because I'm standing here estimating. But if you used the numbers that I actually did, right, it would be 10 to the 3rd. So it would be 1 over 1,000. There's a total of 1,001 molecules, right? If you really think it's 1 out of 1,001.
Yeah.
Right? Because I asked you over the total number of molecules of phenol, how many are dissociated? It would be 1 in every 1,001. So it's 1 so there's 1 deprotonated for 1,000 there's 1 deprotonated for 1,000 protonated. Mm-hmm. But that means of the total molecules, it's 1 in 1,001. That's why that was there. So yeah, it's a math thing. But you should have been able to answer the question if you knew the approximate, right? Because there was only one answer that even slightly matched it. So, okay. So, and actually here, I wouldn't even be accurate because this has a pK of 9.8 as I have it here. So I'd have to figure out exactly what that turns out with mathematically. Which is why I gave whole numbers in the quiz. Okay. So you don't have to think about it that much. Okay, so the lysine side chain, again, the pK is about 10.5. So again, it's going to be approximately 1,000 of the protonated now compared to the deprotonated. And you notice when we give the pKa, HA, or the acid that we're giving on this side, in this case, is the protonated form of lysine. The pKa of this molecule going to the negatively charged species is in the 30s. Okay, so it's a completely different question. Arginine is more basic, and so it has a pK of around 13. So it actually, so this is the free arginine, this is the protonated form. So the reason for this is, as you can see here, is because of the fact that you have charge delocalization due to resonance, right, for arginine. And so the reason that I show these is that these all have ionizable groups, and they're really critical in charge-charge interactions. So these are some of the things that make salt bridges. Right? If you were going to think of a salt bridge, you're often going to see them between arginines and aspartates or glutamates, or lysines and aspartates and glutamates, right? Because at pH 7, this is going to be in the positive form, and this is going to be in the negatively charged form, right? And so you're going to have a charge-charge interaction. Okay, so I just wanted to point out in terms of how you approximate a PI of a peptide, or anything where you have more than 2 titratable groups. So you can estimate, a true PI has to be calculated. It's a much more complicated calculation to get the absolute PI, and really probably experimentally identified, right, by titrating in the pH. But you can get kind of an estimate of the PI by looking at the two pKa's that govern the formation of the neutral species. So if you have, so if you have an even number of charges, for example, as you do here, right? You have, pK, so if you look here, there are 4 different charges in this molecule, or there are 4 different groups in this molecule that are ionizable, right? There is the terminal amine, there is the lysine amine. Both of these are basic, so at pH 7 they're going to be positively charged. And then you have the terminal carboxylate and you have this carboxylate, both of which are acidic. So at pH 7 they're going to be negatively charged, right? So which 2 groups are going to control, everything else being even, as we get towards the PI, which 2 groups are going to be the most likely to control the PI? Well, we have to have a positive and negative charge that are canceling one another out, right? It's going to be the 2 that are closest in pKa, where one of them is going to be on the positive side and one of them is going to be on the negative side, right? Because those are going to be the things that are zeroing in on the PI. So in this particular case, that is 8 and 4, right? This is going to make a negatively charged species. This would make a positively charged species. So the PI for this is going to be approximately 8 plus 4 over 2, or 6.
Did you say you should call this to pH or pKa?
Did
you say, like, how did you choose the charges again?
So you choose it, so you think about so remember that when we're doing PI of a protein, we're thinking about it, it's going to be in the physiological range. So one easy way to do it is to say at pH 7, right, what's going to be negatively charged, what's going to be positively charged, because those are the charges that are going to cancel, right? And then think about which 2 are the closest to one another in pKa, because if you have, that are on either side of that, right, where one is going to make a negative and one's going to make a positive. And those 2 are going to be the ones that if you average their pKa's, you're going to get the approximate PI, okay? And you can see that actually here. So this is actually what the book, so the book doesn't show you how to do the calculation, which is part of why I'm showing you. But this is actually the titration from the book. They show you this. And you can see that at, as you go up, so if you are really, really acidic, right, you're going to be +2, right? Because both of your positive, because both this and this are going to be positively charged, and these 2 are going to be neutral if you're really acidic, right? That's kind of how they show it here. If you add more base, you'll eventually lose this charge before you lose this one, right? So you're going to go to here. Actually, you lose this charge before you lose that one. Then, eventually, you'll deprotonate the carboxylic acid, and then you'll be neutral. Actually, you'll deprotonate both carboxylic acid, You start out with 2 plus charges. Eventually, as you add more base, you deprotonate your carboxylate, you're at +1. Eventually, then you deprotonate the other carboxylate, now you're neutral, right? Then you go into being more basic, you're going to deprotonate your nitrogen, and now you're at -1. Then you're going to deprotonate your other nitrogen, and now you're at -2. Okay, and if you look at what I approximately calculated as the PI, that's exactly where, if you look at the actual titration, the molecule is. And what I did is I took the 2 pKa's that were closest to one another, but on the other side are making a positive or negative charge, right? Because they're the ones that are, and what I mean by closest to another is closest to one another and closest to neutral, okay? So not just the closest, and which makes sense because that they would have to have close pKa's, right? And so those are the 2 that if you average them, you end up with a PI of the protein. Does that make sense? Question?
Yeah, so if the PI is, say, 10, then would you choose, so can we go back to the last slide, please? So would you choose, like pKa was 10.5 and the pKa was 4? Like if we set the PI to a different number?
You can't set the PI to the different number. The PI is intrinsic in the protein.
Oh.
Right? The PI is a matter of it is the pH at which the molecule is neutral on average, right? In this case, I'm giving you an even number of charges, right? So the average is a little bit more, so in this case, we're actually showing a neutral total molecule. but the solution of molecules is neutral on average at the pi of the protein. It is intrinsic to the protein, right? It is intrinsic to the pKa's of the groups on the protein. It has nothing to do, but what I'm saying is one way to sort of think about what's going to be the charge of the protein is to think about what are the ionizable groups and what kind of charges would they have at pH 7, right? Because that gives you an idea of what's going to be negatively charged and what's going to be positively charged so that you can think about how they are going to balance one another out. Does that make sense?
If for whatever reason we chose the pH to be 9, did we use pKa from 10.5 because they're closer?
Okay, let me repeat this. It has nothing to do with the pH you choose. Pi is intrinsic to the protein. The Pi is the pH at which the protein is neutral.
Okay.
I'm pointing out, thinking about, so the reason I say think about what is ionizable at pH 7 is because I think it's easier to grasp what you use to calculate the Pi if you can think about what the charges on the individual amino acid side chains or the termini are going to be, right? And so it's just it's a good reference point to think, oh, what's negatively charged in the molecule, what's positively charged in the molecule, what's balancing. And so I think it's a good reference pH to think. It has nothing to do with the Pi itself. It's just a good reference pH to think about the charge distribution in the molecule.
Can you just like explain why you picked the
Okay.
for pKa?
Yes. So if we think about the charges in this molecule, right? And so here it's shown in a kind of funny way because these are shown as being, everything is shown in the HA form, right? And that makes sense because we're giving the pKa's of them. So everything is shown as their conjugate acid, right? But if we think about what's going to be negatively charged and what's going to be positively charged as we think about Pi, right? This is why I say it's good to imagine it at pH 7. This is positively charged. This is going to be eventually negatively charged. This is going to be negatively charged. That's going to be positively charged. To approximate the Pi, you have to pick the positive and negative charges where the pKa's of the HA that would create those groups are the closest together. Because that's going to be the tipping point between being a positive molecule or a negative molecule. And halfway between those, you should presumably be approximately neutral. Does that make sense? So if you think about that, the two pKa's of things that become negative are pKa 3 and pKa 4. The two pKa's of things that are positive are pKa 8 and pKa 10.5, right? If we think about the negative and positive pKa's that are closest to one another, or the pKa's of the acids where HA is positive, right? And the bases where A minus is negative, if that makes any sense, that are closest to one another, those pKa's are going to be 4 and 8, right? Because 4 is closer to 8 than 3 is, right? And 10.5 is closer to 4 than 8 is, right? So if you think about it, these are my two bases. If you just do the cross comparison, my two bases, my two acids, which are closer? 8 and 3, 8 and 4, 10.5 and 3, or 10.5 and 4. The two numbers closest in number are going to be 8 and 4, right? So the average of those, so that 8 plus 4 is 12, you divide it by 2, it's 6. So that's how you get approximate Pi. Does that make sense to everybody? Okay. And the whole point I said about the pH 7 is it's really, this is shown in the HA form, right? So sometimes people forget like what becomes negative, right? Because it's being shown as the acid, right? Because that's appropriate for the pKa. When you give the pKa, you're giving the pKa of HA, right? So this is shown in the HA form. And so my point is, if you're shown something and it's in the HA form where everything's protonated, think about at pH 7 what's going to change, right? What's going to go from positive to neutral, what's going to go from neutral to negative. Because that will help you figure out the charge balance to figure out what cancels.
So hypothetically speaking, if this polypeptide was in a solution of pH of 6, you would have part of it be neutral, the other part be positive.
Absolutely. So at pH 6, on average, both of these would be protonated and both of these would be deprotonated, and the net charge would be 0, right? And in reality, every molecule will probably have a slightly different charge state because it's all in equilibrium, and you'd have a distribution, and the overall distribution would be net 0. right? Okay, so on that note, predict the charge state of this peptide at pH 7. So this is the peptide, and let me just point out the pK of this is about 6, of this is about 3, this is about 3 or 4, and that's about 7 or 8. This is actually probably more like 8 or 9, about 9. Okay, so the charge state of this peptide at pH 7, this one is about 6.6, right, almost 7. The pKa of this is about 3. The pKa about this is about 3-ish. And the pK of this is somewhere in the 8s. Okay, so presuming that. So this is your extra credit. So think about it. And I drew it as the neutral molecule. I want you to tell me approximately what is the charge state. How charged is it going to be? Right? Yeah, so presume this is about 6.6, 6.5. Okay? So presume that this is about 6.6 or 6.5, that this is so this is approximately 7. Let me put it that way, right? The pK of the protonated form of this is approximately 7. Okay? The pK of the protonated form of this is not shown in the protonated form of histidine, but the pK of the protonated form is approximately 7. The pK of the protonated form of this is approximately 4, right? The pK of the protonated form of this is approximately 8-ish. And the pK of the protonated form of this is going to be approximately, again, 3 or 4. Okay? Is there anything that can come off of that? This is a charge state. Yeah. If a hydrogen is coming off of isoleucine, we are in deep trouble. Okay? Because then we would have formed a carbanion. We would not be happy. Especially in water. So. Yeah, I showed you the deprotonated form, but I'm saying presume that the protonated form, the pK is approximately 7. Give or take. For this calculation, presume it's approximately 7. Just this is a back of the envelope, approximate what the charge state is of the protein, right? So, whole number approximation, one significant figure, nothing complicated. Okay. Okay, so overall it's going to be approximately -5. Why? Right? So I'll go through the reasoning. Why? The term and I cancel at pH 7, right? Carboxylate, positive charge, they're pretty much gone at pH 7, right? Asp is about -1, because if you think about it, it's the pKa is significantly low, that this is going to be an approximately -1 charge state, right? Histidine is going to be about positive 0.5. It's going to be about 50% protonated. The pKa of the protonated form is almost exactly, it's not quite, but it's approximately 7, which means it's a 50-50 charge to uncharged, charge to neutral, right? So you can approximate that charge as plus 0.5. So you have you have something where all of this is going to be deprotonated, but half of this is going to be protonated. So it only cancels out about half of that negative charge. The overall charge state is going to approximately be -0.5. Make sense? Okay. You have a question-ish.
So if histidine has pKa of
Give or take approximately 7.
Yeah, like more than 0.5, then would it be?
The pK of histidine is 7. That doesn't change. That's an absolute number, right?
Yeah,
That's
like,
nothing.
okay.
If protonated histidine was more acidic, right, then this would be -1. Like if the pK of histidine was like 3, right, protonated histidine was 3, this would be -1 charge state.
Then charge state would
If
be-
the pKa of histidine, and note, the pKa of histidine is a number, and it can change with the local environment having to do with the peptide, right, because of changes in that, but it's an absolute. It doesn't change with the pH or anything like that. It's an equilibrium constant. You know, the pKa is an equilibrium constant in standard state, right? So it's not changing with other things. But presuming that the pK, so if for some reason histidine was now like amazingly basic, this would then be neutral, right? Like if the pK of this was like 9, this would be approximately neutral, right? But it's not. This is half and half, approximately, right? So it only cancels out about half of the charge of that, so that's why it's -0.5. Okay. So now let's talk a little bit more deeply about the different amino acids and what the classifications of the question.
Sorry, going back to the last slide. So, basically it's 50% protonated because it's neutral.
Because the pKa is approximately 7-ish. It's 6.6, rounded up to 7, it's close enough.
Okay.
Yeah. And we're going to go through, all of the amino acids and talk about their chemistry a little bit more, and I think it'll become a little bit more obvious when we get to the histidine slide. Okay, or actually, you know, another place to think about that, I'm just going to go back really quickly, is right here, right? So the pK is about 6.6 at pH 7, and let me just point out, on this slide, all of the things that are in these boxes are the state of this at pH 7. And you notice for histidine, I have both of them circled. That's because it's approximately a 50-50 mix, right? When pH is PI, when pH equals approximately, when the pH is approximately pKa, right? The concentration of A minus over HA is about, you know, you're about at 50-50 for protonated to deprotonated, when the two are equal. Okay, and this is, give or take, that's pretty close to 7, right? Okay. So going back to our classifications, so we have various types of amino acids, and I'm going to go through, this is another way of looking at the classifications. They're acidic, they're basic, there are some that are a little bit acidic but aromatic, that are a little bit, here's histidine, it's kind of partway between all of these, right? Can act as an acid and a base. You have aliphatic, you have polar, you have sulfur, you have flexible and inflexible. So we'll talk a little bit about these one by one. So let's start with the aliphatic amino acids. So these are nonpolar hydrophobic residues, okay? What do I mean by that? So alanine, where that's a CH3 group. So the R groups are right here. So the core is in the box, and the R group is what's coming out above on the stick figures, right? So alanine, where this is CH3. This is isoleucine, right? This is leucine. This is methionine. So methionine has a sulfur group in it and a CH3 group. And this is valine, where this is now an isopropyl group, right? This is an isobutyl group, an isopropyl group, a methyl group, and a, so this is, well, that. The isoleucine is a 1, 2, 3, 4. So this is actually the 2-butyl group, right? So it's actually put on the second carbon of a butyl. If you think about this coming off the other chain, right? So what do these all have in common? They're very hydrophobic, okay? They don't like water, they're very hydrophobic, they're greasy. These are hydrophobic side chains. The sulfur in methionine is generally considered to be unreactive, okay? It has very little chemistry that it actively does. I won't say that there's none, but there's very little chemistry for that sulfur. Okay, so it's generally an unreactive molecule. It's basically a grease ball, much like the others. These residues are often found in hydrophobic interfaces. So when we get more into structure, you'll find that they're often on the insides of coil proteins, and we have things called the leucine zipper, right? Which is because you have a hydrophobic interface that actually helps to keep coil proteins together. Okay, so you often find these in helical bundles, where as we'll get to alpha helices, these can actually come together in tertiary structure, in sort of more complicated tertiary structures, where now your helices will have hydrophobic patches that will connect one another. Okay, and so hydrophobicity of these is actually very important, especially, again, you see things like leucine zippers, and the hydrophobicity matters. Okay, so aromatic amino acids, you have phenylalanine, okay, you have tryptophan, and you have phenol. Now, phenylalanine is very hydrophobic, right? It's a phenyl group. That's it. Now, tryptophan is still very hydrophobic, but you notice it has definite sort of pi stacking. It has a permanent dipole in it, right? Unlike phenylalanine, tryptophan has a permanent dipole. That actually doesn't involve, that is actually something that affects its ability to interact with cations and anions in kind of pi stacking interactions and doing pi stacking interactions, and you do see that with tryptophan. And in fact, it also has a hydrogen bond donor here, okay? Phenol has both a hydrogen bond acceptor and a hydrogen bond donor because you have an OH group. And again, that means that there is a permanent dipole to this aromatic ring. And it can do hydrogen bonding. So tryptophan and tyrosine both have polar head groups or polar groups to them. They can participate in pi stacking, as can phenylalanine, but these can participate much more. Because they're already a little bit more polarized, right? As well for tryptophan, it's got 2 rings, right? So you can think of just the ability of electrons to move. Tyrosine is a weak acid. We've already gone over the pK. It's approximately 10, much like phenol, right? But that means that there is acid-base chemistry that it can sometimes engage in. And actually, although we won't really talk about it in this class, tyrosine can form a radical, and that actually also has implications for proteins. Okay, so there are some times where tyrosine actually is part of radical reactions in proteins. And as you might remember, those radicals are kind of more stabilized because they actually have the ability to be delocalized, right? So I should also say that both tryptophan and tyrosine can be post-translationally modified. And so that means that things can happen to these amino acids after they're made into proteins, and there are enzymes that do that chemistry. So you can get C-monosylation of tryptophan, and you can get sulfation, phosphorylation, and glycosylation on tyrosine. And we'll go over more about what those actually look like later in the semester, but I just wanted to point out that both of these are actually able to be modified. And tryptophan is the least frequently occurring amino acid. And there's a reason that I tell you this, and it has to do with the fact that both tyrosine and tryptophan are very strong chromophores. And because tryptophan is one of the least frequent amino acids, it's often used as a probe for structure, okay, because of the fact that it's rare, and so you don't have it, so you can actually tell exactly where it is, right? So you can use it as a diagnostic probe in some cases for structure. So if we look at the UV absorbance of these particular amino acids, you'll notice that phenylalanine doesn't absorb that well, tyrosine absorbs pretty well, and tryptophan absorbs the best. Okay, and typically, you can see that here. So at 280 nanometers, which is where people often take UV vis spectra for proteins to determine protein concentration, right? What you're really looking at is tyrosine and tryptophan, and this is commonly used to actually determine protein concentration, much like the A260 is used to determine DNA concentration or nucleotide or nucleic acid concentration. And in fact, one of the things, if you are creating a DNA prep and you want to make sure that it's not contaminated by protein, one of the things that you do is you do what's called an A260 over A280, right? Because what you want to determine is that your nucleic acids are pure and are not contaminated with protein content. Okay, so you actually use this to determine protein concentration pretty commonly. Phenylalanine also absorbs in the UV range, but if you look at the extinction coefficient for this, if you look at the quantum yield, it has a much lower lambda max, and its quantum yield is much smaller. So it's not as good. Okay, so and the extinction coefficient here is much, much lower. So it's a very weak absorber compared to tryptophan and tyrosine. Okay, so that's our aromatic amino acids. So next we're going to go on to our basic amino acids. So the interesting thing about these is we call them basic amino acids, but actually they're all positively charged at pH 7. So can these act as bases at pH 7? No, right? If you look at them, they're all hydrogen bond donors. By the Bronsted-Lowry definitions, they're actually local acids, right? They're actually acids. They're hydrogen bond donors, not hydrogen bond acceptors at pH 7. So even though we're used to thinking about amines as bases, and we call them basic amino acids, in terms of like whether they would donate a proton or accept a proton, they would donate a proton, right? Not accept one, because they're protonated. We're not talking about the free base form, we're talking about the protonated forms, right? So the pKa of lysine, which is this one right here, so you notice lysine has a chain of 4 carbons terminating in your amine. The pKa of lysine is approximately 10. Arginine, which has this guanidinium head group right here, is approximately 13. Again, this is resonance localized positive charge, and that's why this is such a good base, why the pKa of it's so high. And histidine, here I have it as 6.5. It's in the book 6.5, 6.6, approximately 7. It's usually taken in that range. It has what's called an imidazole head group, shown here as the imidazolium. Truthfully, this is approximately 50-50 at neutral pH, right? Which means that it can act both as a hydrogen bond donor or an acid and as a base, right? And that dual property of it's actually really important. So I want to point out that the pKa's that are shown are all for the protonated forms of these amino acids, right? Because remember, we're showing the pK of HA, okay? So I have a question. At physiological pH, is it possible for lysine to be a good nucleophile and react to form methyl lysine? Have an idea? Is it possible? How many people think yes? How many people think no? I'd say, come on, it's okay. Either way, there's no judgment here, okay? This is an exercise. So it looks like the no's have it, right? No. Actually, yes, because let's think about this, right? One of the things that I'm trying to point out is the difference between equilibrium and possible. I said possible, right? I didn't say, is it going to go super fast if it's protein? I said, is it possible? Let's think about that. A pKa is an equilibrium constant, right? And if you react it with something else, it pulls it out of the equilibrium. Okay, Le Chatelier's principle in action. Remember that? Okay, so if we think about this and we approximate the pKa's that I had on the other page, about 1 in every 1,001 molecules is deprotonated, right? 1,000 of them are protonated for every 1 that's not, right? But that 1 is a reasonable nucleophile, okay, and can actually react. More than that, I should point out that in cells, lysines can be acylated and methylated post-translationally. In fact, they're really, really important epigenetic modifications as part of the histone code. Okay, they're part of what actually helps to control whether or not our genes are turned on and off. Okay? These reactions are catalyzed by enzymes that, as part of their enzymatic mechanism, have a localized base that actually helps to deprotonate the lysine, which makes sense, right? Because that would catalyze the reaction by making it a better nucleophile. So, just so you know, arginines can also be methylated. So even though it's not something that's likely to happen very often, with catalysis, with a localized base to help pull it off, you actually can get it to go. And I just wanted to point out, that's why I put possible. Possible. Not likely, but possible. And the answer is yes, because remember, if you can pull that equilibrium forward, you only need a few molecules to go, right? And then all of the rest of it will follow. Okay, so getting back to this issue of histidine. Histidine is indeed, it is the only amino acid with a pKa close to physiological pH, which means that it can act as both an acid and donate protons and as a base, because it's got a lone pair here that can accept the proton, right? So it can go back and forth. It is ambidextrous, so if you think about it in this form, in the unprotonated form, the nitrogen with a hydrogen atom is an electrophile, right, hydrogen bond donor, and the other is a hydrogen bond acceptor. Right, so it has the ability to be a hydrogen bond donor and acceptor, depending on what form it's in, and even within the same molecule, it can donate a hydrogen bond, right, and accept a hydrogen bond. And it exists in two tautomeric forms. So we show it typically drawn in one, but realistically this hydrogen could be on either of those nitrogens, right? There are two tautomers of it. Because of this ambidextrous property, the fact that it can act as both a donor and an acceptor, an acid and a base, you often see histidines in catalytic active sites of enzymes, right? Where local acid or local base catalysis is really important. Okay, so this is very, very common. It's often part of the catalytic triad in proteases, among various other places. So histidine is commonly seen in catalytic active sites because of this property, because it can have both charged states, right? Which means it can cycle back and forth really easily at pH 7. Okay, so those are our basic amino acids. What about our acidic amino acids? So our acidic amino acids are negatively charged hydrophilic residues. Aspartic acid, which is here, has 1 carbon, and it has a pK of about 3.9. Glutamic acid has 2 carbons and has a pK of about 4.2. At pH 7, these molecules are negatively charged, which means that even though these are acids, right, can they donate a proton at pH 7? No. Right? They can accept it. So they're actually Bronsted-Lowry bases, which is kind of a funny thing because we think, oh, they're so acidic. But what that actually means is at physiological pH, they're not good acids because they've already given up their proton. They're negatively charged, right? It's like saying a chloride ion can act as an acid, right? The chloride ion cannot act as an acid, neither can the carboxylate, right? Because its proton is already gone. Yeah, these are Bronsted-Lowry bases at physiological pH, right? Because they're no longer Bronsted-Lowry acids. An acid can, by definition, donate a proton. These protons are already gone, right? At physiological pH, their protons are out of there, right? So you're left with the conjugate base. Okay, so what you see at physiological pH is A-. It's the conjugate base, right? So these are hydrogen bond acceptors. Notice the red arrows. These are hydrogen bond acceptors at physiological pH, not hydrogen bond donors. Okay. And I want to point out again, the pKs are for the protonated forms of these amino acids. I'm just pointing out that they're always for the protonated forms. Okay. So what about, just for one more minute, I'm just going to say about polar amino acids. These are neutral at pH 7. We have the amides, and we have hydroxyl groups. So these are asparagine and glutamine. You can notice that these are just the amide form of aspartate and glutamate. Unlike those, these are now neutral, right? Because the amine doesn't get protonated or deprotonated. The hydroxyl groups are on serines and threonines. These are polar. All of these are polar, but relatively unreactive. They can all act as both hydrogen bond donors and hydrogen bond acceptors, okay? And I just want to point out that asparagine, serine, and threonine, so these three right here are all found as post-translationally modified. So this actually, N-linked glycans are actually off of asparagines, and O-linked glycans are off of serines and threonines. And since I work in glycosylation, this is actually something I'm very familiar with. And serines and threonines can also have sulfation and phosphorylation on them as well. Okay? Okay. So we're going to stop here. And Rohini is going to give the next lecture, starting with the next set of amino acids to finish out our amino acid foray.